Okay, the last active transport mechanism we're going to talk about is vesicular transport. Okay. And what this is basically the cell itself will move or deform itself to actually pull things into the cell or kick things out of the cell. Okay. And vesicular transport can basically, it is bulk transport. Okay. To get large things in. When we talked about transport before, we're talking about like moving like little glucose, or talking about moving some sodium and some potassium. But there's larger proteins and stuff that will be moved into the cell, and typically the cell has to like move itself around them. Okay, and so when you do this, the cell, here's our cell. Okay, and let's just say we have something funky out here. And the cell, what's going to do is it's going to grab that funky thing. It's going to grab it. And eventually what's going to happen is it's going to have a compartment with that funky thing sticking inside of it. And this is called a vesicle. Okay, that is called a vesicle. All right. So we have two forms of this. We have this called endocytosis, not endo, okay, which means it's going to pull it inside, and we have exocytosis, is outside, to move outside. So endocytosis, I'm going to pull things into the cell, and exocytosis, you're going to spit it out of the cell. Okay, two important transport mechanisms, especially when you're talking about proteins and like snot. Okay, like the mucus of snot, that's an exocytic process. You know, and your cells will eat things during endocytosis. Now, how do you know this is an active transport? Because anytime you need to move your damn cell, okay, that takes a lot of energy. Okay, a lot of energy, because you actually have to deform the elements inside of the cell. Okay, so we've got a couple of these. Okay, so the first of these, we're going to call it receptor mediated. Okay, and we're going to talk about endocytosis pretty much, because exocytosis is just exocytosis. So on the endocytosis front, I'm just going to draw an arrow. So here we have what's called receptor mediated endocytosis. Receptor mediated endocytosis. So what this is you've got a plasma membrane. Okay, here's our plasma membrane. And they have some divots. You have some divots on them. And on these plasma membranes, okay, they have receptors. Okay, that's what these things are. These are receptors. And remember, we talked about receptors, and we talked about receptor mediated. Is they're specific. They will only bind certain things. And so what they're looking at is they're looking for a substance. They're just sitting there waiting for a substance to bind them. And when that substance binds them, let's just say it's substance diamond. It doesn't matter what it is. Okay, let's just say substance diamond binds to it. What this receptor does is it latches onto it, and eventually what's going to happen is the membrane is going to change it's going to start changing its shape. It's going to change its shape until eventually the membrane will engulf this diamond shaped thing until it's inside of the cell. And that's receptor mediated endocytosis. Let me draw the other receptors just so it's not confusing. Okay, and that's receptor mediated endocytosis. Now you're like, well, how does the energy? You know, where's the energy for this? You know, you need ATP to do this. Well, we're going to talk about non-membrane bound organelles. And you have these things that attach to the plasma membrane. It is called the cytoskeleton. Okay, the cytoskeleton, and it attaches, and you have these things called microtubules and things like that, and you have this stuff called actin and myosin, and it moves. It moves, and in order to move something, you have to give it energy. So the thing that drives this is giving energy to the cytoskeleton so it can warp the plasma membrane itself. Okay, and that's it. So it's receptor mediated endocytosis. Okay, the number two example is we have pinocytosis. Pinocytosis. And basically what this is the vesicles form and they pull in water or they pull in small molecules. I like to think of it as cellular drinking. Okay. Not like going to the bar and drink, but like consuming water. So if you have a little cell right here, and you have a bunch of H2O sitting over here, H2 over here, and what to do with the cell is going to be like thirsty, and it's just going to start forming these little vesicles, and eventually what's going to happen is these little vesicles are going to have water trapped in them. And then they're going to eventually go into the cell. And so you're going to have these little pockets of water inside of the cell. And that's what pinocytosis is. Okay. That's it. Cellular drinking. Again, how does it use energy? It uses energy to bend the cytoskeleton in order to do this. Okay. And then finally we have probably the most important, phagocytosis. Okay, this is cellular eating. Okay, it's phagocytosis. You're going to want to remember phagocytosis because this is how a lot of your white blood cells work, especially the macrophage itself. Okay, so let's just say we have a cell here, a big old macrophage. It's a macrophage. Macrophage, well, first of all, it's not macrophage, it's phage. Okay, this it cleans out a lot of debris. Like if you ever get injured or you have like a chronic infection or something like that, this is the guy that helps clean everything out. And so macrophages, they really don't need to be told what to do. So we're just going to draw a little bacteria around here. And if he sees this little bacteria, the macrophage is going to be like, aha, food time. Okay? And it's going to start moving toward this bacteria. And then what's eventually going to happen is the macrophage is going to change its shape to where now the bacteria is going to be like now inside part of the macrophage. And then eventually what's going to happen is the macrophage will actually contain the bacteria within itself. Okay, and then what the macrophage is going to do is it's got these little things, these little vesicles inside of the macrophage that are going to be like, it's kill time. And they got all these digestive enzymes in here, inside of here, these digestive enzymes, these peroxidases. And what they're going to do is they're going to fuse to the new vesicle, and they're going to kill the bacteria and digest it and break it down. So that's phagocytosis. Basically, it's cellular eating. I mean, it's literally cellular eating. The cell just comes up and wraps around it and engulfs it to where it will be digested and broken down into its component parts. All right. So with that, so we got receptor mediated endocytosis. We got pinocytosis, which is basically phagocytosis, but with water and smaller particles. And then which is water drinking, and we have phagocytosis. This is cellular eating itself. Okay, now if we have exocytosis, let's just do that for completion's sake. Exocytosis. Okay, and it's just the opposite. It is 100% the opposite of endocytosis. So if you've got a cell, and let's say on the inside you have goo. Okay, and goo is usually green. Okay, we'll just make it green goo. And let's just say this green goo needs to be released out of the cell. So what's going to happen is this vesicle, and that's what this is, it's still a vesicle, it's going to be pulleyed. Okay, there is a pulley system across the cells, and it's going to be pulleyed to the cell membrane. Okay, and in the pulleying system, that's going to use energy. That's where the ATP, that's where the active transport of this thing comes from, is I'm using ATP in order to move this vesicle to the cell. And so what's eventually going to happen, what's eventually going to happen is this cell, the vesicle is going to, oh, that's way too big. Don't need it that big. The vesicle is going to move to the cell, to the cell membrane. And energy is going to be expended in doing this. And then what's going to happen is that vesicle is going to fuse with the membrane. And all the green goo, that's blue goo. No, that's green. That green goo is going to be ejected from the cell. Just like that. And that is exocytosis. I'm taking something from the inside of the cell and I'm moving it to the outside of the cell. And it is an ATP mediated process, just like endocytosis is an ATP mediated process, because I'm changing the shape of the cell, and then you're going to use ATP to break it down, and it's a huge energy expenditure for endocytosis and exocytosis. Okay, so with that, we are now officially finished with active transport. We're finished with the plasma membrane, and how we get things across the plasma membrane. All right. So next, what we're going to do is we're going to start looking at the organelles of the cell, the little organs, and we're going to start with the non-membrane bound ones. We're going to talk about the cytoskeleton, and we'll be talking about the ribosomes. Okay. All right. So that's that. Take a break before you continue. Definitely. There's a lot of stuff here.